thanks for the presentation. And based on what Vicente just presented on what are the topics of your talk, so this one will be more focused on maybe infrastructure and how we apply also genomic data analysis, and we integrate phenotypic information for downstream analysis. I will be focusing on rare diseases, but just give also at the end a very short introduction on the application for cancer. As you will see, the platform can be tailored a bit on different use cases. So just as a background, and as Vicente mentioned, I'm from the CNAG, the National Center of Genomic Analysis in Spain, that is co-funded by the Spanish government and the Catalan government, and we give service to research institute and also hospital in Catalonia, Spain, but also in Europe, and we are participating in several European-funded projects. This is a bit of the program that I will be following for this webinar. So basically, I will give a very brief and short introduction on rare diseases to understand a bit more why all the development of this infrastructure. Then I will go in more detail into the RD-Connect Genome-Phenome analysis platform, which is the tool that we have developed, and different modules that can help for downstream analysis and handling of a large number of data sets. Then I will explain a bit on how this development, this tool has been applied in different European projects, and our experience, and what were the outcome of that. And also an overview on other GPAP instances that are not the RD-Connect GPAP, but that are used in other environments, in hospitals, or to share data across regions. Finally, as I mentioned, and if the video works, I will show you how we have applied also as a backbone of this development for the cancer use case and somatic analysis. So regarding rare disease and genomics, so I don't know if you are familiar with the rare disease use case, but rare diseases are defined by the one that affect less than 1 in 1,000 individuals. In total, there are over 7,000 rare diseases described. So they are individually rare, but altogether they affect around 7% of the population. And it is known, or it is established, that between 70 or 80% are of genetic origin. And this is why the genomics really have an important role to play in the diagnosis of rare diseases. Diagnosing rare disease patient is not easy, and indeed this is an infographic from EuroRDis where they show what they call the diagnostic odyssey, where the patient have to go and visit many different clinicians and experts, and also once they want to obtain their molecular diagnosis, it's also not always straightforward. So this is why there is a big need on trying to make all this path shorter. And the scientific community is really working toward that, because having the molecular diagnosis for the patient will help them to, of course, have the proper treatment if existing, but also for clinicians to understand how a patient should be managed, to know about the prognosis of the disease, have access to genetic counseling, and also, of course, for families to reduce anxiety and uncertainty of what's happening. So with all this background, and as we mentioned, rare diseases are rare by definition. So this means that in each hospital or even regions or countries, we still have for each individual disorders very few cases to confirm diagnosis, very few samples available for research, and also for clinical trials. And this is why rare diseases were kind of the use case pioneering the importance of data sharing, and maybe that first enter on all these data sharing infrastructures, and trying to develop a solution so that patient data can be securely shared across Europe or internationally. Data sharing as a concept is necessary, but it's also challenging when we want to do it. Basically, because it's sensitive data, genomic data, and phenotypic data. We have the GDPR regulation, or we also have each country that might adapt it and have different regulations for this type of data. The data might not be standardized, so complicated to share, need some kind of harmonization prior to it. We have different type of data, have genomic data, phenotypic data, other types of omics data. The data is big and is getting even bigger now with exome and genome sequencing that can be done more routinely in hospital environments. And also we are not always incentivized to share, so it requires an additional work that all the people involved need to put in. So this is why several years ago now, 7 or 8 years ago, the RDConnect European project was founded to help this data sharing, and one of the infrastructure developments there was the RDConnect Genome-Phenome analysis platform. So this platform is basically an online system that will facilitate the collation, sharing, analysis, and interpretation of integrated genomic and phenomic data. And this started with the rare disease use case, basically, as I mentioned, for diagnostic and also gene discovery. So as you can see in this little schema, basically the phenotypic data will be submitted in a standard way that I will show you later on. And then the genomic data will also be submitted with the raw files plus the associated metadata. The data will be processed by a standard analysis pipeline in the system and made available for the users to access and perform the tertiary analysis. In this case, depending on the project and upon approval, the raw data, the FASTQ file and BAM files can also be archived at the European Genome-Phenome Archive. And basically, to date, we have over 30,000 Genome-Phenome datasets with more than 350 contributing groups and 700 registered users. All the data in the system that is shared is pseudonymized, and it becomes available to all the users after an embargo period of 6 months. So you have 6 months where you are the only one accessing your own data, and after this 6 months, it gets shared within the whole community. This platform is also securely audited periodically by an external company. So here there is a short video explaining the overall concept, and after I will go into more details. Vicente, please let me know if you can hear the sound. I think here is just music, so that's fine, but...
Yes, we can hear it.
Okay, great. Yeah, so that was a short video, so you have basically the overview I gave in a more fancy way, let's say. And now I will go into more details on the different parts so you can see what can be done and how we harmonize all the information. So first, we collect the clinical data, the phenotypic data. And here, as we are in a rare disease use case, basically we use the human phenotype ontology, the orphanet database, and OMIM for the genetic and molecular findings. And we have a user-friendly interface so that clinicians or the people that are in charge of uploading the data to the system can access. This interface has been created based on the data models from Genomics England, where they have already defined 20 groups of disorders to try to map all the types of rare diseases. So we have used all that work and information to stratify disorders in our system. And we have also mapped every of these 20 group of disorders with the European reference networks, and this was done in collaboration with the Orphanet and the INSERM. So basically, once you select, as for example in here, the template of tumor syndrome, you will see which ERNs might be more interested in this type of disorder or entering this information. And then you will have the list of disorders and the corresponding HPOs that are proposed to fulfill and enter deep phenotyping for these cases. This way we help for deep phenotyping and also to do differential diagnosis, because then the symptoms that might or might not be present are also listed. Of course, if all this information is already available in internal databases, you don't need to go through a manual intervention, and all this can be automatically imported into the system. These forms can also be tailored for specific use cases with controlled vocabulary, so that, as you will see later, it can help to create cohorts of patients to perform downstream genomic analysis. This is how the overall of the information enter look like. So you will have access to all the patients that you have entered in the system. You can filter by the different data enter and also export the information. And among different formats, one that we are currently using and adapting is and have adapted is the GH4GH Pheno packets. So this way, the data does not get siloed even in the RDConnect GPAP, but you can export it to whatever other system or use it in whatever other pipeline tool that you would like to use clinical information for maybe filtering or prioritizing your data outside of this kind of systems. This is the landing page when you access the genomics module. So now we have had a look at the phenotypic module, and now we'll go for the genomics one. So here, as I mentioned, FASTQs have been submitted and the data has been processed through a standard analysis pipeline. So what you get in here are the result of the variant calling and the annotation. So you get your annotated variants in the system, and this is what you would be accessing. And from here, there are different types of analysis that I will very briefly describe. The first one will be the case analysis. So you can analyze an individual or its the entire family, but it's really for a specific diagnosis of that case. And then we'll go through the cohort analysis and the search across all, which is what data sharing enable in this kind of system, that you can create specific cohorts to try to identify maybe a new disease-causing gene, or if a new gene has been described or published, you can very quickly check all your database if there is any of your patients that have a pathogenic variant in that specific gene, so you can diagnose and do reanalysis in a quicker way. We'll also see how you can do patient matchmaking outside of the system. So you have the information within the GPAP, but you can also query other databases internationally. And finally, we have the option of the clinical report that I won't enter into this today. So regarding the case analysis, what the platforms enable is a user-friendly system to access the data from whatever family structures you have. And then based on the annotations, you can filter the genomic information, prioritize it, and link out to many of the different databases that are used for variant interpretation. What the system also offers, and this is a work that we did in collaboration with the EGA, the European Genome-Phenome Archive, is to store the data, as I mentioned, the data can be stored there. So we had the BAM files from the SolveRD project, which were around 20,000 datasets that were stored there. So when a user wanted to identify a candidate variant or a variant of interest, they could request the BAM or the CRAM slice from that data that was stored at the GA. And then through the G4G HTS GET protocol, they got back and they had the rendering of the alignment so that they could visualize in real time the genomics alignment to validate or not a candidate variant. Then now I will show you the use case on really browsing the overall system, so all the data that you might have available in here or in a specific instance of the GPAP. Basically, you will be able to create cohorts based on the ownership. So you can look only at your samples, your group, specific European reference networks, or the RD Connect data. You can choose the origin. Then you can create your cohort based on the control vocabulary and the ontologies with what you have, the one that you have used to submit the clinical information. So HPOs, ORFA, or MIM, but also the sex, the affected status, if there is already a causative gene identified, there is consanguinity or not, etc. And finally, you can create the cohort based on metadata, on the file's metadata. You want to only look at genomes, at exomes, or specific kits from an exome, etc. Once you define your cohort of interest, that might be with a specific symptom that you think might be relevant to try to identify a new disease-causing gene, then you can use the annotations to filter the data, and you would get a list of the genes and the variants that have been identified and in which cases have passed your filters. If you identify an interesting variant, then you will be able to see to whom it belonged. You will have a summary of the HPOs of that case, even if it's not your case. So you can see if it fits or not your hypothesis. And if it fits what you were looking at, or if you think require more information would be of interest, then you have a contact button that will automatically send an email to you and the submitter of that case to put you both in contact. And you can start a new collaboration, and this would be done outside the RD Connect GPAP, and this is really a scientific collaboration that can be done and start from here. The next level of data sharing would be outside the system. So what we've seen until now is really, for example, within these 28,000 data sets. But now imagine that you don't find whatever you're looking at, or you would like to identify other patients, then the system is connected to different patient matchmaking options. We have the beacon, but also a matchmaker exchange, which is what I'm going to explain now. And for matchmaker exchange, the question that you are making is if there is any similar patient, meaning that with a similar phenotypic information and also a similar hypothesis on the gene that you are looking at in other databases. And you can see that there are many nodes connected internationally. You have Phenome Central, GeneMatcher, the Broad, Decipher, etc. So within the system, we are connected to different endpoints. And then on the left, you can see, for example, one of our participants with a candidate gene and a description of the symptoms with the HP ontology. And this way, by querying the other system, you will retrieve information of similar patients. You will get a score between 0 and 1. 1 would be a perfect match, meaning exactly the same symptoms and exactly the same candidate gene. And otherwise, because you get the score, and you can see the phenotype associated to those similar patients. If you think you find something interesting, again, you can click on the contact, an email is sent to both you and the submitter so that you can start a collaboration from there. And yeah, this slide is also to pinpoint that after the diagnosis part, we are also starting to work on trying to avoid a second odyssey with the treatment. So we work within Solvar-D and now in Herdera, that I will explain later, on the Tritabulum database that aim to associate variants, genes, and treatments. And what we've done is to connect to this type of databases so that once you identify a candidate or the pathogenic variant or diagnose your patient, quickly you could access information on the literature, on which treatment are currently given to those patients or have been described basically for now in publication. This is mainly done because now many centers with maybe are not expert in a specific disease, and by doing exome sequencing and genome sequencing are diagnosing patients that they might not know about the treatment. So it's really to try to give all the information at once. Okay, so now I have explained the platform, a bit the different modules and the analysis capabilities. And what I would like to show you very briefly is some of when or how we have applied the system in different projects with real data and with real use cases. So the first project that we used the RDConnect GPAP to take to gather or collect all the phenotypic and genomic information is Solvar-D. So Solvar-D was the base of one of the aim of this project was to reanalyze unsolved cases, so cases with an exome data that is negative after going through the whole routine and hospital workflow. Because, as you may know, around 50% of cases are still negative after the analysis that are performed. So basically, the idea was to collect 19,000 data sets with the phenotypic data also included, and to do a reanalysis to try to diagnose some of them. Of course, this is part of the project. There were other parts that I won't enter into details in generating new mixed data, etc. And what we did in Solvar-D was to build or connect different infrastructures that were existing together to have a whole workflow enabling this massive reanalysis of data, and to automate everything so that it would be possible. So basically, the data was entering through the RDConnect GTAP and the phenotypic module Phenostore that I presented, and then it was reanalyzed also with different approaches and different clusters from the other places. And as I mentioned, the data was distributed but also stored for long-term archival at the EGA. And here is just one slide to explain the results. So these are results from the reanalysis of the first 6,000 cases that have been recently published. And thanks to the systematic reanalysis and all the infrastructure that was set up within Solvar-D, that also involved many groups for the analysis and for the interpretation, the European reference network, they managed to solve 800 previously negative cases. And when we calculated the amount of time invested by the experts to do this reanalysis per variant that was proposed as candidate variant, it was a bit less of 5 minutes. So basically, we managed to create a reanalysis workflow that could work in a semi-automated way so that it was possible for experts to go through all this information and manage to solve all these cases. And now we are scaling up, and Solvardi will be part of all the infrastructure and development that were done in Solvardi will now be also part of Herdera, which is the European Rare Disease Research Alliance. I won't enter into details of this big project and alliance because it's really huge and we need a lot of time. I just want to explain a bit of one part, which is within the clinical research network, where basically we are going to scale up what was done in Solvardi and also going towards a federated approach. So this is what I would like to show you now. So we have different work packages with the aim of reanalyzing and analyzing what we call distributed data. So to centralize again the data in one place and do this reanalysis, but also to go towards federation. So the data do not need to move the country, but basically it's the same approach to harmonize all the genomic, phenotypic, and now also other omics data, RNA-seq and long-read data set that have been already sequenced. All this to provide to the Herdera ecosystem in a uniform way so that the analysis task forces and the interpretation task forces can work together to try to solve cases. And then there will be also new omics data that will be generated and reanalyzed to complete and be integrated in those analyses. And basically here it's where all of this is to mention that for collecting this data for the distributed approach, for the one that we are still centralizing during the first year, we will be using more or less the same system. So through the RD-Connect GPAP for the phenotypic, the metadata, and the genomic data. And once we have all the information harmonized, processed with the same pipeline, then it will be distributed to the Herdera ecosystem so that it can be further analyzed and interpreted. But basically it's to show that the system can handle hundreds of thousands of data sets to be harmonized and put together. And here, this slide is to illustrate this part of we will be starting from this distributed approach, centralizing and uploading, moving the data here at CNAG, but the idea is really to move to a federated approach so that all these reanalyses can be done locally through different systems, either other GPAP instances or whatever systems available, and then federate everything so that we can talk to each other. And going towards federation, this means that we would need to have several instances, and this is something that we have been also doing here at CNAG. So we started with the RD-Connect GPAP, the one that now I have described and explained, but we have also instances that can be installed in a unique hospital. So this is the work that we've done, for example, in a hospital in Navarra here in Spain. Can be instances where different hospitals from the same region in here, Catalonia, have an instance to share their data. Well, here is the use case of the cancer that I will also show you a bit later. But basically, what we've done also in Spain is through a project called Impact, which is meant to create a cooperative infrastructure for providing, for helping diagnosis with complex genomic technologies. So we have built a system using different nodes of the GPAP for the analysis and interpretation of the data. The workflow is very similar to what I presented. Here, the clinical data is not in our system, but it's in another system that is connected to the different GPAPs through programmatic interfaces. Then the data gets sequenced, processed, and uploaded to one of the nodes. We have 3 different nodes in this project, one in Catalonia, one in Navarra, and one in Galicia, which are different regions from Spain. And the idea and what we are working towards is really to federate those nodes, meaning that independently of where the data has been sent, either in Catalonia, Galicia, or Navarra, they can see each other data and make these questions queries to see if there are other patients with similar phenotype and genotypic data and genomic data associated. There is another project that also tries to have all the raw data in a federated EGA node, where they would be able to reuse the data for research purposes, and they are working towards that. So this is not yet a reality. And yes, so these federated use cases that we are developing in Spain has also been was first done within the 1+MG framework with the proof of concept of the rare disease use case. So here I put the link to a demo where you can see the overall workflow of a clinician or a geneticist accessing a platform and wanted to have more information on a genomic variant that is in another system. So how they could federate the different systems and give access to the information, but basically within the overall tools that were put together to manage to do this use case. The GPAP was used for federating these tertiary analyses and querying other systems. All of this was done using several existing J4J standards that were very useful to standardize everything within the different parts of the workflow. And finally, I come to the cancer part. So here it's a video explaining a bit how this kind of development and all the technology that is behind for managing complex data and big data sets can be then applied to other use cases, such as, for example, cancer. So I think my next one is a video. And Vicente here, let me know if you don't hear it, because then it might be problematic, because here there is the voice explaining everything.
According to the World Health Organization, about 1 in 5 people will develop cancer in their lifetime. Cancer is caused by genetic mutations that lead to the uncontrolled growth and spread of abnormal cells, known as tumors, which disrupt body functions. Accurate detection of these mutations enables precise diagnostics and tailored, personalized treatments, which are key to improving outcomes and saving lives. Thanks to recent progress in genetic sequencing and analysis technologies, these genetic alterations can now be reliably detected. However, data interpretation remains a major challenge. Identifying cancer-driving and treatment-relevant mutations among thousands of genetic alterations requires not only expertise, but also the integration of clinical, genomic, and cancer-specific knowledge from multiple sources. This is a complex and time-consuming process. The Cancer Reporting GPAP is a user-friendly and comprehensive state-of-the-art clinical decision support system designed for next-generation sequencing, cancer data interpretation, and reporting. It's powered by advanced big data scalable architecture technologies, enabling real-time queries. This allows the users to extract insights efficiently, even from large and complex genomic datasets. The system provides a secure collaborative environment that can be installed on-premises or in the cloud, ensuring flexibility and compliance with data security standards. It can use common standard variant files as input, including those generated with the pipelines available in the Cancer Analysis GPAP. Our solution simplifies and enhances the interpretation process by integrating a diversity of oncology data sources, enabling automated variant filtering and prioritization, and facilitating efficient molecular report drafting for both hereditary and acquired cancer. The cancer reporting GPAP includes an automated classifier of oncogenicity for several types of genetic variants, including single nucleotide, small indels, copy number alterations, structural variants, and gene fusions. Unlock powerful cancer insights with cancer reporting GPAP. Comprehensive data integration. Leverage genetic and clinical data for accurate interpretation. In-depth tumor insights. Gain detailed information on mutational burden, tumor signatures, homologous recombination deficiency, and genomic instability. Automated variant prioritization. Identify potentially oncogenic and pathogenic variants with high confidence following international guidelines. Advanced data exploration. Easily filter, browse, and analyze patient genomic data using multiple annotations and customizable queries. Pharmacogenomics and treatment insights. Highlight pharmacogenomic markers, treatment recommendations, and clinical evidences. Customizable reporting. Generate comprehensive, tailored reports to support informed medical decision-making. Would you like to know more about the cancer reporting GPAP? Contact us at...
Yes, basically, this was to show how we can adapt the system to different use cases. And that's the end of the presentation. We are happy if you want to collaborate either by submitting data if you have genomic data that is of your interest. If you want to join the Herdera Research Alliance, it's also open, and here you have the email to coordination. Or if you think that other possible collaborations are feasible, so this is free for academics, and here the email if you want to contact us. And I think I will finish to thank the team of engineers that have developed the system, and also the different projects and many people involved in all of those that have used and enabled the development of the system.
Great. Thanks a lot, Leslie.
I think I will stop sharing so we can see each other. Yes.
Yes. So now we can start